Uh, all right. Hello, everyone. We're going to be looking at the regulation of gene expression today. I'm going to share my screen. Okay. Can you guys see it? Okay. All right. So this is kind of parts of the beginning of genetics and evolution, this unit that's kind of important in biology and on the USABO. So first we start off with kind of what gene expression is, right? Kind of a review. Gene expression follows kind of this central dogma of biology where we have DNA being transcribed to RNA, and then from RNA we can translate into protein. This is what cells use to create different proteins, different molecules that they need to survive. It's also how genes are expressed because in the end, the genes have the DNA sequence, which translates to the protein structures. So there are ways to regulate what proteins are produced by the cell, and that's what we're going to cover today. So one way to regulate gene expression is through restricting transcription. The organisms that are able to regulate transcription of genes can benefit by saving the resources or materials for other tasks, and some products may not always be needed. One way to do this is through use of operons. Operons are sections of genetic materials that can be split into multiple smaller parts and rely on feedback loops and other proteins to regulate the transcription of genes. Pictured on the bottom left, the blue bar is a simple diagram of the regions within an operon, which we'll look at in more detail later on. And an operon in action is in the bottom right. Notice the different proteins attached to the DNA, as well as the chemical structure that's interacting with the red block over there. Okay. So here we see a bit more in detail that simplified diagram from the previous slide. There's three main sections of an operon. There's the promoter region, the first part, and followed by an operator region, then genes after that. The promoter and operator regions will control whether regulation is active, also affects the transcriptions of genes further down the DNA. Certain proteins have specificity for the sequence on some of these regions, and then they will bind there if possible. For example, polymerase will bind to the promoter region of an operon, where it is free to move along the rest of the DNA and eventually transcribe the genes at the end. But it can also become blocked by a repressor protein, and the repressor protein will bind to an operator region. Repressors are often activated or inactivated by presence of some molecule or chemicals. One way to remember the order of the parts in the operon is through the mnemonic Prague, which is a place in the Czech Republic, and it sounds like the acronym PROG. And that's from a Bozeman video, which we'll look at later. But yeah, on the slide, you can kind of see brief descriptions for some of the parts in the operon, as well as the two main proteins there. The arrows kind of point to where they bind to, as well as where they're going. For example, in polymerase, it binds to promoter region and can move on along the rest of the operon. Repressors, yeah, they attach to the operator region, which we'll look at later. So there usually are two operons that are commonly taught in classes as well as the campus. First one's, or one of them is the trip operon. This deals with the control over tryptophan synthesis within cells. By default, this operon is in the on position, which means basically that tryptophan can be produced by transcribing the genes on the end of the operon through protein synthesis as well. Uh, so in the absence of tryptophan, then the polymerase is able to bind to the promoter region, then moves down the genes, and then it's able to make RNA from that. By using a negative feedback loop, the cell can stop using resources in tryptophan production as long as the tryptophan is now present in high enough quantity. So this means that the tryptophan molecule will act as a co-repressor and they activate repressors. Thus, the repressor will then bind onto the operon and block polymerase from transcribing the genes. And because of that, tryptophan is no longer produced. And remember, this happens when there's enough tryptophan within the cell. Right. So the feedback loop is if you don't have tryptophan, polymerase is able to transcribe genes that encode for making tryptophan. And once you have enough of tryptophan, then it binds back to the repressor and then it starts blocking the polymerase from making more. Uh, and yeah, because of this, the cell can conserve its resources and not have to continuously create tryptophan when it's not needed. Another operon is the Lac Operon. This one's a bit different. It targets a different mechanism and also has uh, different ways of functioning. Lac operons will control the creation of lactase, which is the enzyme that's used to break down lactose. Remember that the terms that end in ace are usually some sort of enzyme. This one's a bit different as well, that it starts kind of what we call the off position. So the repressor is already attached to the operon. And because of that, polymerase cannot move to transcribe the genes. However, in presence of lactose, the thing that lactase acts on, right? In presence of lactose, it will bind to the repressor and then it will inactivate the repressor and allow for polymerase to move along in the transcription. Right. So this one also uses the negative feedback loop, so it's a bit different, where uh, usually in no presence of lactose, then the operon is blocked, and then when you actually have the lactose, then the cell needs to break it down, so it allows the polymerase to make the lactase. Yeah. So one thing to note between difference between the two operons is lactose acts as an inducer, which will inactivate repressor and allow polymerase to move on in transcription. Whereas in the other operon, the tryptophan will act as a co-repressor and helps to activate the repressor. So in some questions, I think also once in the past that Yusuf has also asked this, but you need to kind of know what the co-repressor and inducer terms mean and what they do. So yeah, the clear example of the differences are from those two operons. Right? Inducer inactivates and co-repressor activates. Um, Moving on, this is kind of a diagram of eukaryotic gene expression. It's a bit different from prokaryotic gene expression because the differences between eukaryotes, prokaryotes. Remember that eukaryotes have those membrane-bound organelles, right? It's a bit more complex than the prokaryotes. That kind of causes differences in the way that genes will eventually be expressed in the cell. So in eukaryotes, uh, it follows that central dogma, but when you create the RNA strand from the DNA, it undergoes some sort of processing within the cell uh, that includes adding on poly A tails, the 5' cap, uh, removal of introns, and then the splicing together of axons. And then after that, it leaves the nucleus. And then when it leaves the nucleus, it becomes known as the mRNA. Before that, it's just a pre-mRNA strand. Then it moves on to translation, which is somewhere like on a ribosome within the cell. And then this is different from prokaryotic expression in that prokaryotes just make the mRNA and then move directly to translation. So on that diagram, like all those steps, you can regulate at uh, any one of those steps. It will start to change what proteins are being made, and we can move on to talk about one of them in particular or a few of them. So one of them is epigenetics. This is how the cell can kind of modify its DNA through small changes, either like activating or inactivating certain parts of the DNA. Epigenetics is unlike random mutation in that the changes can be like reversed, whereas mutations are just errors that start to persist within a genome. Epigenetics usually uh, is a response to some environmental factors. This kind of plays into the thought of nature plus nurture instead of just one or the other. Right, development of organisms, partly because of nature, which is like genes and all the normal stuff, but you also have nurture, which is uh, external factors that may affect development. Another way is RNA splicing. RNA splicing occurs like in the previous slide when we looked at it uh, in the step called RNA processing on that diagram, right? You can see the darker regions on the strand of the RNA are exons, the lighter ones are introns. In RNA processing, right, you can cut out introns and leave just exons there. But you can also regulate like which parts are expressed through RNA splicing, where you have different combinations of exons, and because of that, you're able to create multiple mRNA strands from that one mRNA. Right on the bottom is the kind of a diagram from Khan Academy about that. Uh, as you can see, uh, different variations of that cutting out of introns and then splicing together exons and create different sequences of RNA. And then in protein synthesis, those different sequences translate to different proteins. And because of that, right, you can have multiple different proteins coming from that same pre-mRNA strand. Another notable one that's commonly asked is DNA methylation, acetylation. This is from Campbell's. It's kind of a summary of the process. Right? Usually, the DNA methylation will decrease transcription of certain parts of the DNA or certain genes, whereas acetylation can increase it. So with the methylation, right, we add methyl groups CH4 to the or CH3 to the parts of the DNA, and then these DNA, right, they wrap around the structures known as histones. Then you can also have acetylation that will allow for more transcription of these types of genes. DNA methylation, acetylation, they are also kind of a way that a cell can respond to its environment. Again, nature plus nurture. Then we move on to another topic within the regulation, its use of transcription factors. Transcription factors, they're kind of ways for the different proteins to bind to different regions of DNA. So kind of the key ones we're going to talk about are activators, enhancers, and then there's this protein, I think it's called the DNA bending protein. And then RNA polymerase, which is the one that will transcribe the RNA. Yeah. One thing to note, Campbell always has pretty good diagrams, so this is from Campbell's. But if you need to refer to something and need a visual diagram, usually Campbell's has good drawings of the harder parts. So, but basically, what this thing is saying is that activators, there's certain proteins, they will bind to parts of the uh, gene that are called enhancer regions. They have specificity to each other, right? So then because of that, they're able to bind. And then once the activators bind to enhancers, we have this thing on the bottom, DNA bending protein. Uh, DNA bending protein is there because, well, like the name suggests, it will bend the protein. Bend the DNA. But why they need to bend is because uh, there's a certain there's like two attachment points on that mediator protein thing, which helps the RNA polymerase bind to in the next step. But basically, right, because it bends, and you can see in the diagram, it can attach to kind of the top of the group of mediator proteins. And when we go on to the next one, it also binds to that TATA region on the like beginning of the gene, and because it's kind of like in that bent structure now, then RNA polymerase can bind to the uh, beginning region and begin the RNA synthesis. So that's one thing. That's another like thing we should know about gene expression, right? Sometimes they'll ask questions in the series of steps, and then you kind of need to know what those things are. Like, what are enhancer regions? What are activators? And then need to know how they play a role in the RNA synthesis. Okay. So I'm gonna go back a bit to operons because it's kind of hard to like visualize them without a diagram. That's good. What I drew is very simple, but if you go search on YouTube or like the web, you'll probably find different simulations of operons. They're very useful in helping you to understand like what goes where and how the entire process works. So we're going to go watch a Bozeman video on it because it was pretty good animation. I'm not sure.
Hi, it's Mr. Anderson, and in this video I'm going to talk about the operon. The operon was discovered in the 1960s by three French scientists, Jacob, Louf, and Monod. That's really bad French. But they got to coin the term operon, and basically it means to operate. And so it's mostly found in prokaryotic cells, mostly found in bacteria. There's a few eukaryotic operons, but in general it's just going to be found in bacteria. And so it led to other terms that you've maybe never heard of before, like regulons and modulons and stimulons. Stimulons are groups of operons operating together from one stimuli. And so it just means to operate. That's what an operon means. And so the one that they discovered was the Lac Operon. They were studying this in E. coli. And so the Lac Operon is designed for E. coli to break down lactose. So lactose is going to be a disaccharide. And remember, if you're a bacteria living in our gut, you're eating whatever we're eating. And so as the material moves down through our digestive system, they have to be ready to quickly shift depending on what the food is. If it's sugar, they have to be able to break down the sugar. If it's proteins, they have to be able to break it down into amino acids. And if it's a disaccharide, in this case, they have to move it into the cell and then break it down and chemically digest it. And so they use a Lac operon to do that. Now, this is a picture of Prague, but that's not how you spell Prague. But it is the way that I remember the parts of an operon. You've got a promoter, a repressor, an operator, and then genes. And so if you can remember Prague, it's going to get you half of the way there to remembering how an operon works. And so the neat thing about bacteria is that if they have a number of different genes that are used to achieve a certain task, they'll put them right next to each other. And so as the RNA polymerase moves down the DNA and makes all of the messenger RNA and makes all the proteins, you're just ready to do whatever the job is. And so the genes are going to be located right next to each other, in this case, in the Lac operon, to deal with lactose. What else do we have? Well, we've got the promoter. The promoter is going to be the region of the DNA where the RNA polymerase can grab onto the DNA. We also have an operator, and so think of that like a on-off switch that's going to turn on or off the operon. And then the last thing we have is a repressor. And so this is a Lac Operon. It's PROG. It's promoter, repressor, operator, and then all of the genes. And so what are we trying to break down? We're trying to break down lactose or deal with lactose. Remember, lactose looks like this. It's a disaccharide. But in this little model right here, we're going to represent it as a little pentagon, a yellow little object that looks. And so all of a sudden, a lot of lactose is present. So what is the Lac Operon going to do? Well, it's going to deal with that lactose. And so all of this lactose is moving around, remember, but eventually one of the molecules is going to bump into the repressor. As it bumps into the repressor, it's going to change the shape of that repressor. And so a quick digression, everything promoter, operator, and genes are DNA. So they're going to be part of the DNA of the bacteria. But the repressor is a little bit different. It's a protein. It's a protein that came from a different part of the DNA. There's going to be a regulatory sequence, either upstream or downstream of the operon, that creates the repressor. It plugs in nicely to the operator, but once the lactose fits inside it, it's going to change the shape of that repressor. What does that do? Well, it opens up a region where that promoter can allow the RNA polymerase to get on, and then that RNA polymerase is just going to drive right down the DNA. It's going to make a bunch of RNA. It's going to make a bunch of those proteins. What are the proteins designed to deal with? Lactose. And so they're going to break down the lactose. Now all the lactose is gone. What's going to happen to that repressor? It's going to return to its original shape, and that shape is going to fit into the operator. And so if there's no lactose present, we would say the operator is in the off position because the repressor is activated. It's activated, it's sitting inside the operator. It's physically breaking the movement of that RNA polymerase. The RNA polymerase can't move down and code for those genes because the repressor is sitting in its way. What happens next? Well, lactose shows up again. Lactose would move into the repressor, free it up, and then it's going to be able to make those genes. And I've got a simulation at the end of this video that shows you in a little more detail how that takes place. Before we get there, I want to talk about the trip operon. The trip operon does essentially the opposite of that. So the trip operon is evolved in bacteria to deal with tryptophan, or more specifically to deal with the absence of tryptophan. So tryptophan is going to be an amino acid, and it's required to make proteins. It's one of those 20 essential amino acids. And so the trip operon basically is designed to make tryptophan if it's not present. And so we're going to get tryptophan in poultry, in milk for that matter. There's going to be high levels of tryptophan. But if a bacteria doesn't have tryptophan, there's a number of different genes that are required to make it. And so they can make their own. And so we've got an operon called the trip operon. How does that work? Well, again, we've got PROG. We've got our promoter, our repressor, our operator, and then our genes. But if we have tryptophan present, so tryptophan is going to be these yellow hexagons. If we have tryptophan is going to fit inside the repressor, and it's going to change its shape so that it fits in the operator. And so in this case, if we have a bunch of tryptophan present, then we don't want to make tryptophan. And so the repressor is going to set that operator to the off position. What happens if all of a sudden the tryptophan goes away? If there's no tryptophan in the diet, well, the bacteria is not out of luck because it changes now the shape of that repressor. And so once it's changed the shape of the repressor, RNA polymerase can grab on, drive down that operon, make all of those genes, and those genes can be used to create more tryptophan. And so it's a great kind of a feedback loop. So in a Lac Operon, we're going to turn it on if we want to break down lactose. In a trip operon, what we're going to do is create tryptophan if we don't have it or if it's not present. And I really didn't understand how operons worked until I stopped thinking like a scientist and really started thinking like an engineer. We've got a problem that we have to solve, and one of the great ways we can do that is using an operon. And so I'm going to
Okay. We're not gonna watch the simulation, but I can provide a link to it. So yeah, that's kind of what I have. And then we're gonna have a little quiz thing. I'm not using Kahoot, I'm using quizzes. So I think it's a bit better, but We can get out of the device. Now pull up the quizzes, share the screen, the join code, and then we can work on that. Okay, so you guys can go to that website and then enter the code, and then we can answer like a few questions, discuss them as well. Just again, go to that website, enter the code, and then we'll do a few problems about this topic. Okay, I guess we'll get started. There's no one joining. So, Questions are on your screen, I think. So yeah. Okay. Uh, we're all done? Yeah. Okay. Okay. Good job. Uh, all right. Yeah. Good job, guys. I'm gonna go over the questions because there are two that are free response, so like I can't really set an answer, right? So we're gonna go over kind of what they are, and if possible, I'll see what answers you guys gave. Questions. Okay. Number one was in lack of tryptophan, the uh, I should say trip operon is able to transcribe genes for the production of tryptophan. Is the repressor attached or detached to the operon? Right. So this one's asking about trip operon. Uh, remember the trip operon, uh, the tryptophan production one. So that in presence of tryptophan, you're not going to be creating more tryptophan, right? So when you have tryptophan, it's going to act on the repressor to bind to the operon. So that means, uh, wait, sorry. Anyway, the answer should be detached because the repressors block the polymerase from transcribing genes, and thus it needs to be detached for transcription to occur. Uh, I got confused. Sorry. Next one, two, lactose serves as an inducer for the Lac Operon. It will. Okay, so the answer here was it will bind to the repressor because lactose will kind of change the structure, and the repressor will fall off from the operon. The third one, which types of cells go through RNA processing before translation? This one's the one that we talked about differences between eukaryotes, prokaryotes. Uh, and only eukaryotes need to go through RNA processing, right? That's the cutting out of the introns and then doing all the addition of various parts. And this happens in eukaryotes only because mainly, like when you move from one section to another, uh, if you don't add those modifications, then the RNA might degrade within the cell itself. For development based off of nature or nurture, uh, the answer was trick question because it's supposed to be both of them, right? When we talk nature and nurture, we have genes, and then we different processes that can regulate what genes are expressed, such as the methylation, DNA, epigenetics, what we covered. Right, so it should be both nature and nurture play a role in the development. And last two are kind of around the same on the same topic and gives you a gene. The OPN1LW gene is a gene that encodes for a red cone photoreceptor pigment. According to the UCSC Human Genome Browser, the gene sequence is 14,790 base pairs long. There's one identified mRNA product from OPN1LW. It is 1095 nucleotides long. Why is the mRNA strand shorter than the gene? Uh, so there's this gene has what- there's the multiple exons, right? So when you have exons and you cut them between, then you always have like a fewer amount of introns than exons. But because of the RNA processing, you're always going to take out parts of a pre-mRNA strand because uh, they're the introns and they're not really needed for the translation of that RNA strand into protein. So because of that, you'll have a kind of shorter RNA strand than the original gene sequence. Then last one, it's asking you why is the encoded protein in the end only 364 amino acids long? This is kind of, I guess, tricky if you don't think about it, but remember all RNA or all RNA strands have what the start and stop codons. So because of that, right, uh, one of those, the stop codon does not really play a part in that protein, right? So because of that, your final protein will be shorter, usually one amino acid shorter than the RNA strand because it's not part of that final structure. So yeah, here we're given that there should be, right. If you do the math, divide by 3, you'll have 365 amino acids, but because of that stop codon, then the final protein will only have 364. So yeah, those are the last two answers. Uh, oh no, I can see it. Okay, yeah, something like that. Okay, so those are the quiz. Uh, that's what I had for this lecture. So yeah, thanks for coming. Yeah, bye. Next week we'll have um, lectures on having more genes and evolution will have. Uh, we'll be talking about Mendelian inheritance next week.